So this lesson is really our beginning of exploring plant diversity. And we've looked at plant diversity throughout our entire semester because we've looked at these different plant species and we've talked about different plant habitats a little bit when we visited the prairie and when we visited the oak savanna. But this is really our formal introduction to plant evolution and then the resulting diversity. You had an introduction to evolution in your last lecture. This puts it in a little bit of a context because we'll be looking at the evolutionary history of plants. And in our upcoming lessons, we'll look at a survey of the plant kingdom, and then we'll look at a broader scope when we look at plant communities. Oh, and we'll also spend some time talking about how do we organize and categorize plants. So we'll talk a bit about artificial classification systems and natural classification systems. Our specific learning goals for evolutionary origins of terrestrial plants would include 1. Really defining characteristics of plants. 2. We will list and describe some of the key evolutionary adaptations that we see as plants moved from an aquatic environment to land. We'll look at the key plant divisions that reflect examples of those evolutionary adaptations. And we'll actually spend more time doing that in our upcoming lecture than we do in this one. And then we'll apply the knowledge of those evolutionary adaptations in plants to the creation of a cladogram. So you will be looking at the evolutionary relationships, and you'll actually plot that out in a phylogeny or a cladogram. That's actually going to be one of your assignments for this week's lesson. And as a little aside, I extended the due date on that because I wanted to spend a couple minutes in lab on Monday actually talking to you about that cladogram because I think it is a little bit confusing and you might need a demonstration of that. So although I didn't really highlight this very much, we've been talking about plants in terms of their evolutionary history actually since the first day of class when you were given that big group of 150 plants of Wisconsin. It was organized by their evolutionary history. So if you note, the first group of plants were the seedless non-vascular plants, and then the seedless vascular plants, and then the gymnosperms, and then the angiosperms. That actually reflects when we see them first in the fossil record. The next series of slides, they're actually not that important. There's just a series of pictures, of 5 or 6 pictures of plants, and they're actually sequenced in the order of their evolutionary history. So the first plant we see here is a moss, and then you'll see a fern and a gymnosperm, and then a series of angiosperms. So these pictures are meant to reflect the evolutionary history, but also the great diversity of species that are found in the plant kingdom. The moss plant that you saw in the previous slide, we would consider both seedless and non-vascular. Here we have a horsetail. Horsetail is in the phylum Pteridophyta, and it is seedless. It reproduces by spores, but it is vascular. So here we have a fern, also in the phylum Pteridophyta, but actually not that closely related to the horsetail. They're also seedless and non-vascular. Both the ferns and the horsetails represented a group we call the fern and the fern allies. And during the Carboniferous era, these were kind of the kings of the forest, so to speak. So some of these could be 30 meters tall, and they really created entire forests and were responsible for probably most of the fossil fuels that we have today. So there's fascinating biology with these species, even though most of these big giant species no longer exist. Historically, there's some fascinating biology and ecology that resulted because of these ferns and the fern allies. This slide just shows us some of the sori on the underside of these fern leaves. We'll look more closely at reproduction in the mosses, in the ferns, in the gymnosperms, and the angiosperms in an upcoming lesson. And then in the fossil record, we see the gymnosperms. So the woody plants. This is the next kind of adaptation we see in the fossil record, and it's reflected here by this picture of white pine. And this is another one of the seed plants. This is a cycad. Non-flowering though. In the fossil record, we see the flowering plants, the angiosperms. And this slide just shows us some of the fascinating diversity of the flowering plants. Remember, with the flowering plants, we don't just have the seed, but you have the flower and the fruit, right? So we see some incredible diversity and lots of human applications that are directly related to the fruit and the seeds of the flowering plants. We know that there is this boundless diversity of plants. We haven't even scratched the surface probably of the diversity in plants. For example, we know there's over 10,000 species of moss. Many of these moss species have not yet been identified. And we know there is incredible economic value in plants. So pretty much every part of our economic system is in some way connected to plants, whether it's food or fiber or medicine. Again, vast untapped resources and many ways that we could become more efficient and more sustainable by better understanding plants and understanding plant diversity. So as we think about this plant diversity, the importance of this plant diversity, not just for economic reasons, but also for other sustainability reasons, I'd like us to think for a minute why we categorize all of these distinct organisms together as plants. So we've talked about other types of organisms, like the fungi, for example. How are the fungi and the plants distinct from each other? So I want you to just reflect on that. I have down here a think, pair, share. Obviously, when we're doing this online, you can't think, pair, share. But take a minute and think, well, what makes a plant a plant? How is a plant distinct from all these other organisms? So generally, the first thing someone says when we say, what are characteristics of a plant? They'll say they're photosynthetic. So, and then all the things that being photosynthetic includes. Having chlorophyll pigment, having a chloroplast, etc., etc. I should point out that some plants have actually lost their ability to be photosynthetic. So there are several parasitic plants. One common species that you'll find around here is Monotropa uniflora. It's called ghost plant. This is a plant that actually is able to harvest energy. It's a mycorrhizal species that it's kind of complicated because it is parasitic on a mycorrhizal fungus that is symbiotic in a symbiotic relationship with several different types of oak trees. So it's kind of a complicated relationship. But I only mention that because there's about 300 species of plants that are no longer photosynthetic, but obviously their common ancestors were photosynthetic. So most plants, the vast majority of plants, are still photosynthetic, and therefore they are able to capture sunlight energy and transform that into some kind of a usable carbon source. Plants have roots and shoots. Plants have cell walls composed of cellulose, and we saw one distinction with the fungi. So fungi obviously are heterotrophic. We talked about that in lab. And fungi have cell walls, the true fungi have cell walls composed of chitin. Plants have apical meristems. So if you recall from a few lectures ago, we talked about these points of mitotic cell division where we have new nodes forming. We see these on the roots and the shoots, and of course some plants also have those lateral meristems. Plants must defend themselves without moving. So why do we mention that? We mention that because plants have to have some way to overcome all the different ways that they're going to be attacked. So by herbivores, by abiotic factors, non-living factors, by pathogens, and because of this, because of their immobility, plants have devised a whole suite of biochemical responses or secondary compounds to defend themselves. And that's one of the most kind of important and fascinating and maybe poorly understood aspects of plants, all the ways that they sense and respond to the environment. And part of the reason they have such a suite of ways to defend themselves is because they are in some definition immobile, right? So even though often plants can like shoot out their seeds, right? We talked about the touch-me-not plant that has seeds that can kind of fly out, or attach their seeds to your clothes and move long distances. Really, for the most part of their life cycle, plants are immobile. And this is important because of this immobility, they have devised this big suite of secondary compounds. Plants also have something called an alternation of generations, and we'll see this more in our next lesson. So when we start talking about the plant life cycle, we will talk about the multicellular haploid phase that we call a gametophyte, and the multicellular diploid phase that we call a sporophyte. So now we're going to go back in time about 3.8 billion years to observe the first forms of life on Earth, to early Earth. And I'm just going to warn you, once we go back in time, you should not touch anything or like move anything or go off the path, because you could really, mess everything up. So just get prepared. Make sure you're all ready to go, because we're about to go back in time. So hold on. Doodly doot, doodly doot, doodly doot, doodly doot. That's us going back in time. I hope you're ready. Okay, so we've arrived. We are on early Earth. It's about 3.8 billion years ago. And I went to early Earth 3 billion years ago because this is some of our earliest fossil evidence of life on Earth. So we have these stromatoliths that are evidence of early bacterial life forms. This is some fantasy art by Don Dixon, and Don Dixon, this is his interpretation of what early Earth might have been like. So what was early Earth 3.8 billion years ago? What was it like? Let's take a look at the next slide and talk about that. This is what we believe early Earth was like. We think the atmosphere contained a lot of nitrogen, some water vapor, carbon dioxide and carbon monoxide, methane, ammonia, little or no oxygen. And part of the reason for this, little or no oxygen, is because the core of the Earth is iron. And scientists believe that this core of the Earth was actually rusted, which required the consumption of this oxygen to rust the core of the Earth. So any oxygen that may have been on Earth at its early stages of development was probably consumed to rust the core of the Earth. So in addition to the atmosphere not being conducive to human life on Earth, the rest of the environment was also very intense. So there was meteorite bombardment, there was lightning, there was volcanic activity, there was no ozone, so there was intense UV radiation. By the way, I realize I spelled lightning wrong. Every time I look at this slide, I remember and then I forget. So don't spell lightning like that. So a little question for you. You don't have to submit this in any form to me, but just something for you to think about. Um, I brought some seeds back with me, or brought some seeds with me to early Earth, so I took them on our little time traveling adventure, and they're in my pocket. I want to throw them around Earth. I'm wondering what's going to happen to these seeds that I left behind. I want you to think about that. Are my seeds going to live? Are they going to die? What do you think is going to happen to my seeds that I brought back? Are we going to come back, go forward in time again, and find that plants rule the world? What do you think will happen to them? So you'll recall in the previous slides I said that I went back 3.8 billion years ago. And one reason I chose to go back 3.8 billion years ago is because it's some of our first fossil evidence of some of the earliest life forms that are these cyanobacteria that form these fossils called stromatoliths. So the fossils are formed, here is an example of fossil, they're formed when one layer of bacteria, and every layer of bacteria produces kind of this slime layer. The slime layer gets coated with different types of sediment and dirt and etc. And another layer of bacteria will grow on top of that. That layer of bacteria is covered with dirt, and so on and so on and so on. So after millions of years, we'll end up with these stromatoliths that are evidence of these cyanobacteria colonies. The cyanobacteria are also important because they were photosynthetic. So they may be responsible for helping to oxygenate the earth. We said that most of the oxygen was consumed, in early earth was consumed by rusting that iron core. So these early photosynthetic organisms were primarily responsible for increasing the concentration of oxygen in the atmosphere. The concentration of oxygen in the atmosphere is important because more complex cell forms like the eukaryotic cells, they undergo aerobic respiration and require oxygen for aerobic respiration. Aerobic respiration is important because we can actually acquire more usable energy through aerobic respiration than through other types of metabolic processes. So aerobic respiration was required for more complex eukaryotic life forms, including plants. So now we're going to move forward to about 500 million years ago. And we used to say that 500 million years ago, plants were among the first organisms to move from an aquatic environment to land. So prior to plants being on land, we used to say that there was really nothing else on land. But now we know that the fungi and fungal-like organisms probably colonized land before plants. That's actually, I think, a really important distinction, and it's a really important factor because, for one, we know now that almost every single plant has a fungal symbiont. So some plants have to have these fungal symbionts just to live, to survive. I also think that as we consider things like sustainability, restoration, etc., the unique interactions between plants and fungi will need to be considered much more carefully than they historically have been. And by our realization and understanding that the fungi and fungal-like organisms colonized land first, it gives us a better foundation for understanding how plants and fungi interact, and it gives us a better foundation for understanding the unique diversity of the fungi and their plant symbionts. So there's a lot to be learned. There's a lot of work left to do in understanding the plants and the fungal-like organisms and their symbioses. So let's go back to plants. We know that plants moved to land about 500 million years ago. And the earliest plants on land we call non-vascular, seedless non-vascular. We've looked at some of these. We've looked at, in our common plants list, at the liverworts and the mosses. Another group that you'll hear about, I don't talk too much about, they're called the hornworts. Then in our fossil record, we see the seedless vascular plants like the equisetum, the ferns, the spike moss, the club moss. Then we see the seed plants, the gymnosperms, the cycads. And then finally, most recently in the fossil record, we see the flowering plants, the angiosperms. About this slide. In a few slides, you're going to be asked to make a phylogeny or cladogram of the evolutionary history of plants. And this is actually basically what you're going to be doing, recreating this phylogeny. And so I wanted to point something out. Here is the gymnosperms and the angiosperms. They represent the most recent plants in our fossil record. And you're going to look at several characteristics of the gymnosperms, of the angiosperms, of the seedless vascular plants, of the non-vascular plants. So you're going to look at a series of different characteristics of these organisms, and the organisms that share the most characteristics we are going to assume are most closely related. That makes sense, right? So based on their numbers of shared characteristics, we will create this phylogeny. This phylogeny we can interpret like this. So we know at some point in the evolutionary history of gymnosperms and angiosperms, there was a shared ancestor. So the shared ancestor is represented here by this node. But there was a branch between the gymnosperms and the angiosperms. So they share an ancestor, we see the gymnosperms, and then some new derived trait is apparent in the fossil record of the angiosperms. So then we see the angiosperms in our fossil record. So they share many characteristics, but the angiosperms have what we call derived traits of the fruit and the seed and the flower. And we could say that about each point along the way, right? So the ferns share many characteristics with the gymnosperms and the angiosperms, but we see new derived traits in the gymnosperms and the angiosperms. So they share a common ancestor here, but at some point along the way we see selection for new traits that are apparent in our fossil record of angiosperms and gymnosperms, and so on. So this will make more sense to you, I think, when you look at that plant cladogram activity, but for the most part, you basically have all the answers to that question. It's kind of just a way of thinking about the relationships in, excuse me, in this evolutionary history of plants. So as we're thinking about these phylogenies, we can ask ourselves, what is the shared ancestor to all terrestrial plants? And this is a question we continue to refine the answer to. About 5 years ago, we believed it was a green algae called Chara, C-H-A-R-A, and more recent evidence suggests the closest living ancestor of plants is this Spirogyra. But whatever the case, that's still kind of an ongoing question. Whatever the case, we know that there had to be some pre-adaptation to life on land. So those pre-adaptations may have included adaptation to periodic drying. So if you think about algae kind of being on the shores or the interface between water and land, maybe this algae is going to be able to withstand a receding water table, or being on the shoreline for extended periods of time so it would dry out. Also some kind of adaptation to higher light intensities, so clearly There's less UV radiation that's going through the water, and once it's on land and there's no water kind of filtering that UV, then it will have to be able to withstand that higher UV intensity. When we look at the green algae that we believe are closely related to plants, we know that they share some similarities, and some of these are kind of specific biochemical or cellular similarities. So I'm not going to ask you to like memorize this list of similarities, but it's just kind of to illustrate some of the characteristics that are shared, that we believe are shared between the terrestrial plants and their closest green algae ancestor. So some things that we found in these close ancestors is that they share a rose-shaped complex for cellulose synthesis. They share a peroxisome enzyme to limit the loss of organic products due to photorespiration. They share a similar structure of flagellated sperm. And there is a formation of phragmoplasts. So if you remember when we talked about cell cycle and the formation of new cell wall, we talked about the phragmoplast. So again, this is just to illustrate some of the ways that we might characterize the shared ancestors of plants, and both the pre-adaptations and then the similarities to land plants. The last slide focused on what plants share with their algal ancestors. So what are some similarities between plants and their nearest relatives? Now I would like you to think about, well, what are the derived traits? What's new in plants? So if you remember, I talked a little bit about the derived traits that we saw in the angiosperms versus the gymnosperms, and the angiosperms and gymnosperms versus the seedless vascular plants, and so forth. Now let's think about all the land plants versus their algal ancestors, and what are the derived traits that we see. So what's new in plants? And just take a moment before you go to the next slides and think about what we would have to have in the land plants, what traits we would have to have in land plants that we would not have in their aquatic ancestors. Our land plants are obviously going to have some way to prevent water loss. So if you're an aquatic plant, water loss and also transport of water to all parts of the plant are kind of a moot point. You're in the water. Land plants may want to have some way to remain erect. So especially for plants that, if you're not the first one on land, right, and land is starting to get colonized, you probably want to have some way that you can get the sunlight better than everything else that's kind of laying around on the ground, right? So remaining erect, having better access to the sunlight. And a way to transport gametes and protect embryos. So if you're an aquatic plant or an aquatic ancestor of plants, your embryos are probably, your gametes are probably moving through water. So we see in land plants new ways to transport their gametes and to protect the resulting embryos. So some more specific, those were kind of big picture changes in plants or adaptations in plants in the previous slide. Here are some more specific derived characteristics in the land plants. Apical meristems, multicellular dependent embryos, alternation of generations, multicellular gametangia, spores, a cuticle layer, transport tissues, and secondary compounds. So we'll talk about some of these in more detail. So when we look at the fossil record, this graphic shows us where the major innovations occurred as land plants diversified. So the earliest plants on land would include the seedless non-vascular plants. But in most of the seedless non-vascular plants, we see some way to prevent water loss. So the vast majority of these plants will have cuticle and stomata. Then in the fossil record, we have the early vascular plants. The vascular plants, or the early vascular plants, do not have seeds or flowers or fruit, but they do have vascular tissue for a more efficient transport of water from the roots to the shoots, and also it provides them some way to be upright. We also say that the vascular plants have true roots and leaves. Before we get to the vascular plants, we don't consider the non-vascular plants to have true leaves and true roots. Then we have the first seed plants, and we see in the first seed plants pollen. It's a new way to transport gametes. So prior to the first seed plants, sperm is moved through flagellated cells that move through films of water. And we have seeds and wood in the first seed plant. And then we have the flowering plants, where we obviously have all of these other adaptations in addition to the flower, the fruit, and the seed. So a few slides ago, I commented that it's important for us to note that the fungi and fungal-like organisms probably colonize colonize land before plants, and that's important because it really helps to shape the ecology of plants in a much larger scale, right? So the fungal plant interactions that have been going on for millions and millions of years are important to plant ecology and to sustainability questions and restoration questions, etc. But it's also important for us to think about what occurred after plants colonized Earth. So especially once we have plants that require pollinators, like the flowering plants. So we see a number of highly successful insect groups like the wasps, the bees, the ants, the butterflies, flies, beetles, that evolved in conjunction with the flowering plants about 150 to about 50 million years ago. This is just a little aside here. I'm always fascinated when I see the fossil records of ancient insects, and this is an ancient centipede from is believed to be about 300 million years ago. This is there's a lot of fossils like this in the Smithsonian. So if you've never visited the Natural History Museum in Washington, D.C., the Smithsonian, which is free, by the way, you really should, because it's mind-boggling to see these. To the adaptations that we see in plants, one of the key adaptations and first adaptations we see as plants move from an aquatic environment to land is a cuticle and stomata. So if you'll recall, the cuticle was that waxy outer coating that we see secreted from the epidermal cells. So this helps to prevent water loss. And when plants have a cuticle, they will need some way to, still have gas exchange occur, and then we will have these special guard cells that open and close the stomatal pores to allow for water to move into the atmosphere. We talked about, remember, evapotranspiration and the movement of water from the soil to the roots throughout the entire plant body. So these new features in plants were actually among the very, very, very first things we see in the plants that first colonized land. And next in the fossil record, we see vascular tissue. So vascular tissue helped the plants remain upright, and it helped plants conduct water and nutrients. Now, it's interesting because people that study the plants that most scientists call seedless non-vascular, the experts who study the seedless non-vascular plants actually do not like them called non-vascular because the non-vascular plants actually do have a form of conducting cell. So the conducting cells that the mosses and the other bryophytes have are, and the liverworts and the hornworts have, the conducting cells aren't the same and they're not as sophisticated and specialized as the cells we see in more recent plants, but they are still cells for conducting water and sugar. So I just point that out because it's actually a little bit of a pet peeve for people that study those plants that we call them non-vascular. I'm still calling them non-vascular because 90% of the plant science world does, and that's what you'll see in textbooks. But I'll point out the fact that they obviously still have to have some mechanism for transporting water and sugar, although it is not as complex and specialized as the mechanism that we see in more recent fossil record. Next in the fossil record, we see more efficient ways to transport gametes. So pollen actually contains the sperm cell. Before we see pollen in the fossil record, sperm actually moved through a film of water. So there was still this kind of attachment or requirement for water for these plants because they had to have water to move their sperm to an egg. We'll look at this a lot more closely in the next lecture when we look at the life cycle of moss, ferns, gymnosperms, and angiosperms. And then also in the fossil record next we find seeds, so a more efficient way of protecting the new embryo and also disseminating that embryo. And I don't have a picture of this here, but I should also add that then finally in the fossil record, we see the flower, the fruit, and the seed. So we're protecting the embryo, and we're also interacting with pollinators. And this allowed plants to become very successful. So we see in a few slides from now, we'll look at the species diversity with each new adaptation. And in the most recent adaptations, the flower, the fruit, and the seed, we see kind of an explosion of plants, and they become very important in the fossil record. Or there's a lot more diversity, I should say, in the fossil record. When we consider the evolutionary history of plants, we traditionally break them up into a few different groups, including the bryophytes, the fern allies and ferns, the gymnosperms, and angiosperms. And we attach specific new adaptations to each group. So for example, in the bryophytes, we would say we would find the cuticle and the stomata. And then in the ferns and the fern allies, we would say we find the vascular tissue. And in the gymnosperms, we would find the seeds and wood. And of course, all of the previous adaptation. And then finally in the angiosperms, we would have flowers and fruit and seeds and all the previous adaptations. So this graphic I actually referred to a couple slides ago, and you'll note this is our timeline, so millions of years ago, and here we have the different groups of plants. So this bar, the width of this bar indicates species richness. So the wider the green bar, the more diverse that population or that group of plants is. So one of the things that we can we see sort of a trend here that with each new adaptation, we tend to see a decrease in the species richness of the previous group. So it's best seen here, or it's most obvious here, when we see about 140, 150 million years ago, we have the first fossil evidence of flowering plants. Well, look what happens to the species richness of these other groups, particularly the gymnosperms, right? We see significant decrease because the flowering plants basically can outcompete them in the same habitat. So an assignment that you have that correlates with this lesson is the plant cladogram activity. And cladistics is classification based on a common ancestry. We would call this a natural classification scheme. So we're looking at the phylogeny of these groups. Phylogeny is the evolutionary history for a group of species. And when we're creating a phylogeny, we use as much evidence as we have available. So the evidence could come from other living species, it might come from the fossil record, it might come from molecular and biochemical or cellular data. So there's a lot of different sources of data that we might use to create a phylogeny. Sometimes we have many different sources, and sometimes you'll only have one. For example, we might create a phylogeny of the HIV virus. So HIV is a virus that mutates very, very rapidly. And for the most part, when we create a phylogeny of the HIV virus, we just use genomic data. And we are able to then input that information and determine basically the phylogenetic history of that particular virus. The CDC might do this, in fact, if they're looking to sort of trace the source of an infection. And not just in HIV, but in other particularly viral diseases that mutate quickly. So you guys today are going to look at the evolutionary groups of plants and look at at a group of different characteristics within these plants. So you're going to look at some molecular data, you're going to look at some morphological data, you'll look at some developmental data, and you will look for whether or not a particular trait exists within a group of plants or not. So there's a table that you'll fill out, and in this table you will put either a 0 for that trait being absent or a 1 for that trait being present. So you're going to complete the table, and then you're going to use that table to create a Venn diagram of sorts. So you will group together the plants that share the different traits, and basically it's like you're putting them into these little baskets. If they have the trait, they go in the same basket together. If they don't have the trait, they go in a different basket. And then this diagram will be used to put into a branched phylogeny. So I've given you a very, very simple superlinear phylogeny to practice with, to do this with. And I have a video for you in your additional resources that it's a simple video that kind of walks you through the process of creating a cladogram or a phylogeny. What was I going to say? You may want to, and I think it actually makes sense, and maybe I should have waited for this activity until after your next lesson. You might want to wait until after you finish the lecture on the life cycles of the liverworts, the mosses, the ferns, the gymnosperms, and the angiosperms. The reason I mention that is because this cladistics activity, or the plant cladogram, is actually a bit confusing for students on several levels. So my suggestion, and I've also provided the answer key to some of it in the activity, so you could kind of work on it, see how you're doing, see if you're on the right track, and then see how you can fix what you're doing. I'm also going to talk about it in lab. I'll take about 5 or 10 minutes in lab on Monday, but I would like you to attempt it before lab so you can at least have given it a shot and kind of know what we're talking about when I go over it a little bit on Monday.